Hi everyone, I'm Sehi. I'm currently a postdoctoral researcher at Harvard Medical School. Today, I will present our work that we did to better understand various genomics data visualization workflows. Genomics is a field of study that studies DNA, which includes the blueprint of key molecular building blocks of life. This helps us understanding evolution, population diversity, and human health, which lead to important medical applications including precision medicine. However, the analysis of genomics data is challenging for several reasons. It has a large data space to explore. Also, there is a large number of specialized data formats. Given these characteristics, people commonly use specialized visualizations for their analysis. To motivate our work, these are some of the common genomic visualizations. On the left, it shows sequencing information in a linear layout with stacked glyphs. On the center, four matrices show genomic contact information. And on the right, structural variants of breast cancer patients are displayed in a circular view with connected edges. As a visualization researcher, an interesting aspect in genomics is that there are many specialized visualizations that reflect on users' diverse analysis needs. However, oftentimes it is inefficient when users have to switch between different specialized tools over time in their workflows. Our long-term research goal is to understand how we can build a tool that can satisfy diverse users' needs. One step toward achieving this goal, our objective in this paper was to identify various genomics visualization workflows. Moreover, we wanted to identify different challenges that users confront when using existing tools in the workflows. We also wanted to come up with implications that can guide us for future research. For achieving these objectives, we conducted semi-structured interviews with the domain experts. The study lasted about 1 hour for each participant. The 9 study participants were senior scientists, postdocs, PhD students, and software engineers who regularly analyze and visualize genomics data in their work. During the study, we asked several questions to identify participant workflows and then asked challenges that participants confronted at each stage. This is one example of workflows that we identified. It starts with the data processing and analyzing using an R package. The result of the analysis is then explored using a GUI-based tool to validate their analysis. After navigating to numerous genomics coordinates of their interest in the visualization, the participant captures screenshots and uses Keynote to add simple arrows and labels to highlight their findings. This is then presented to their collaborators to get feedback. Frequently, they had to go back to previous stages to make changes and conduct similar tasks again. After reviewing the workflows of all participants, we found several consistent patterns. We identified that people commonly switch between different working environments for a single workflow up to 5 different ones, including Python, R, and cloud services. Also, people tended to use more than one visualization tools for a single workflow. Overall, people tended to use GUI-based visualization tools for quick exploration and then use low-level libraries to see overview or make presentation quality visualizations. Lastly, the overall workflow was heavily iterative and repetitive. For example, one participant manually navigated to 10 different genomic coordinates for a single dataset and then repeated these manual interactions for tens to hundreds of datasets. Overall, we observed that the integration between data tools and visualization tools were not smooth, making the overall workflow ineffective and even challenging. For example, several participants analyzed large datasets in a private server, but then they had to send files to local machine since they did not find an easier way to visualize them without sending files. Another issue was that none of a single visualization tool satisfied both the creativity support and learnability. This resulted in using a GUI-based visualization tool for quick exploration of data, and then using low-level visualization library to create publication quality images. These are some of the takeaways from our study. Participants largely appreciated the GUI-based tools for quick and easy visualization, but they were commonly dissatisfied with the lack of flexible customization. In this aspect, one common limitations for those tools were that adding simple visual annotation were not possible. Given the interdisciplinary field of genomics, I think it is highly desirable to enable better communication and presentation. Given that genomics workflows involve many context switching between tools, supporting better automation in their visualization workflow seems to be important. This includes visual encoding, data manipulation, and user interaction that can be applied to many number of data sets. Lastly, people with computational skills highly appreciated the ability of Jupyter notebooks to record and reuse the same analysis and visualization pipelines for the future analysis. I think studying a way to combine key strengths of GUI tools and computational notebooks will enable much more efficient genomics visualization workflows to help a broad range of genomics people. In summary, we conducted user interviews to identify various workflows in order to gain insights on how we can fulfill a wide range of users' needs. With that, I would like to thank all for listening to my presentation.